Okay, so we are here. We got lesson 5 group problems. Time to go over all of those. So the first 4 problems are very, I don't want to say repetitive, um, but they are kind of asking you to do the same type of things over and over again. And it's a lot of steps. So it took me a long time to do everything. But here we go. So let's go over everything. So on this first question, Margaret Green's doing the study. That's very nice. She gets all of these different um, phenotypes in the generation. So the first question that we have here is determine the allele arrangement in the heterozygous parent. So in order to find that, you have to find out which two of the phenotypes of the progeny are the most represented. And so if you look at this, the numbers that are biggest are the 76 and the 69. So the two parent arrangements are going to be the agouti, wild type, and mahogany. And then wild type, ragged, wild type. So there you go. The next question is determine the gene order. Now, our reading said to make a guess as to which one is accurate or which one is sorry, let me back up. So in order to find the gene order, you have to know what the result of a double cross will be. Essentially, when you have 3 genes, the middle one will be flipped. So what I did is, even though I wrote down here make a guess, I actually kind of thought ahead of my mind. So I know that these are going to be the smallest representative because they're the smallest ones. So what I did is I would compare these two, the largest ones, against these two, the smallest ones, and I'd see what one gene change would make these statements true. So for instance, I said agouti, wild type mahogany. Well, which one of these has 2 of the same? So here's agouti, um, ragged, wild type. Well, 2 of those are wrong. Or sorry, hold on. Wild type, ragged, wild type. Okay. I'm sorry. Let me double check to make sure that I'm doing this right. So my guess, I was ram. So that was ragged, agouti, mahogany. Okay. So if we do that for this first example, we would need a wild type, agouti, mahogany. Oh, yeah, there we go. That's right. So I was sorry about that. So yeah, if we look at this, with this genotype, we have agouti wild type mahogany right here, wild type mahogany. So that's the same. The agouti is just different. And right down here, wild type ragged wild type, we have agouti ragged wild type. So we know that in both of these, from the smallest to the largest, it is the agouti that is different. So we know that's going to be middle. I put R on the left and M on the right. It doesn't really matter which one. But just because that's how they were listed earlier, that's the ones I picked. So if we come down, so you notice on both of these, the bottoms are all the plus for the wild type because that's what the purebred wild type parent is going to be giving. So just to kind of write this down and make sure. So I guessed RAM, and then I took this one. So it would be the wild type for ragged, agouti, mahogany. So you'd get right here the wild type, an M, and you flip the A. So wild type, mahogany. There's that. And the same thing where if you flop the agouti, you get ragged, agouti, wild type for there. So that's where I got it. And looks like I typed in weird numbers. These are both 1. I'm going to have to double check those. In my answer. So do these match smallest group? Yes, they do. So that is the correct order is RAM. Construct linkage. So for this one, what you have to do is take two genes. So for instance, right here, we're going to take R and A, and if there's a crossover between R and A, what are the four possible genotypes? So if you had RAM, the possible ones would be R, wild type, or you could also have a double crossover and you would get R, wild type, M. And then there's also the opposite genes that you'd be crossed over with. So if we start out with our first result, sorry, hold on. This one up here. So if we go wild Goudy mahogany, it's right here. Wild Goudy mahogany. If we cross them both over, or yeah, if they're both flipped over, I mean right here, you would get all wild type, and that's where we get the 16. And then the opposite one, they would be all the letters. The M could also double cross, and that's how you'd get the 1 and the 1, which is also the smallest phenotype. You add all the numbers together, 16 plus 18 plus 1 plus 1 is 36. Take 36, you times it by 100 and divide by the total progeny, which is 206. And that will give you, let me put that equal sign in there, that will give you 17.47. And that's the number of map units that are between R and A. And you just do the same thing over again for A and M. So these bottom two are going to be the same because it's a double crossover. And here we're just switching the R and the A. So you would start with M, and then it's normally wild type A, but you flip them and you get R, wild type A, wild type. 12 and 13. Similar stuff as before, 12, 13, 1, 1. That gives you 27. So 27 times it by 100 and then divide by 206. And that gives you the number of map units, which is going to be 13.11. And I made a little drawing, and that's it. So A is going to be in the middle, R and M with either 13 or 17 map units. Calculate the interference. So what you do is divide the map units by 100 and times them by each other. That's the first step. So I do that, and I get this. It's 0.022. You times that by the number of progeny, and this is the expected number of double crossovers, so 37. And then you divide the actual by the expected, and you subtract that number from 1. So that would give you the interference percentage, which is 94.6. And that's all the questions for problem 1. This one's going to be very same thing. Another chart of all the different variations and all of the offspring we got from it. So first question, determine the gene arrangement for these 3 linked genes. So the parental chromosomes, which are going to be the 2 most popular ones, SC plus, S and V. So that's one of them, SC plus, SV. The other one is SC, S plus, V plus. And there's that. So the next one, determine the gene order. So kind of the same thing. If you come down to the minus, so like let's see right here, SC, SV. Well, that one shares 2 with this top one. So it's only SC that's swapped. So we're thinking SC should be in the middle. So let's check here. So if we did, or flipped just SC, does SV plus, or S plus V plus match up here? S plus V plus. Oh, and there's SC. So yep, that works. We know SC is going to be in the middle. And so we can just do double crosses if it was. So if we take SC or S, SC plus, and V in that order, and we flip the SC, you get S, SCV, and then the 3 pluses down here. And those match if you look again. So we know that's right. Next question, same thing as we were doing in problem 1. You list the 4 possibilities. This time we're going to be doing S and SC. And so we start there. These 2 match up there. But then if the last one was not double crossed, you'd get the 150 and 156 from up here. And that is the results. Those are the 4 possible chromosomes you can get if there's a crossover there. So add all the numbers together, 330. Divide 330 by the total progeny, which is 1,000, and then times by 100 to get a percentage. Or not a percentage, but the map units. So you get 33 map units. And we're going to have to do the other thing for SC and V. So these 2 were already presented. They're the double crossover. They're going to be there. So then if there's just one other crossover, it's just going to be this S that's changing. And if you come back up here to the numbers, 46 and 30, 46 and 30, add them all together, you get 100. 100 times 100 divided by 1,000 is 10, so 10 map units. And there's my pretty map of it. I haven't made it proportional, as you can see. So that's 30 and that's 10. It may not be super accurate, but there we go. In the interference values, the same thing. You have to divide both these by 100, each of them, and times them together. So you get 0.03, and you're going to times that by the number of progeny to get the expected, so 30. And then the actual of the double crosses. So let's those two. 10 plus 14 is 24. So 24 divided by 30 equals 8. 1 minus, or excuse me, 0.8. 1 minus 0.8 equals 0.2, or 20% interference value. Number 3, same type of stuff all over again. Determine the gene arrangement. Last two phenotypes are called stripe and glassy. Okay, so that note was just for me. I'm actually going to move that up here because if I remember working on this, I got confused because there's only 6 phenotypes shown, which means 2 of them are at 0. And so that was just my note that coal, striped, and glassy are not listed on here. So we come down here. Parental chromosomes are going to be the 2 most populous ones. So coal body, plus plus. I put the pluses. It's not with the coal body. I just know they're wild type. I don't know the order because there is no order given up here. And then glass eye, striped. That was the other big one. So now you have to find what the opposite is. So, or for the 2 smallest ones. So the 2 smallest ones are the coal striped and the glassy. So here we have a coal body and a striped. So coal is the same. This one wild type is the same. So it's just the stripe that changed. Let's check this other one in the stripe. So glassy is right. There is a wild type. But then striped is here, but not there. So we're going to know that striped is in the middle. And that's just basically what I did there. There's that and the opposite one, and those do match the smallest ones. So that is correct. Create a linkage map that shows the distances. So same thing, you got to find the mapped units. So the 2 double crosses had 0 representation. So we just take them out. And then if we go with a large one, so coal body, we know that if we do, what should I do first? Yeah, CNS. And so if these both change, but it's only a single crossover, you're going to get all 3 of the mutation. That's going to give you 29, and 27 for the opposite one, where they're all 3 wild type. So I had 29 and 27 and 2 zeros. You get 56. 56 times by 100 divided by 1,000. That gives you 5.6. So 5.6 map units apart. So it's pretty close. Same thing over again. You're going to get 2 zeros. And if we just do the cross the other way, you're going to get this wild type, S wild type, and then C wild type G, and that's going to be 11 and 8. So add those together, so you get 19 times 100 divided by 1,000, the total progeny, 1.9. So super close. And draw the map. There you go. Interference value, 5.6 times 1.9. Oh, you got to divide them by 100 first. That's going to give you this teeny tiny number, like 0.001064. If you times it by 1,000, number of progeny, we're expected 1. Well, what's 0 divided by 1 in junk? Now that's 0. What's 1 minus 0? Oh, that's 1. So 100% interference rate. Next one, more recessive genes, and all in here. And right here it says all phenotypes not mentioned are assumed to be wild types. So I'm going to be doing the pluses for wild type again. So what's the parental genotype? You get the bractic, the fine, and then the anther in the other one. Everything else is the wild type. So we'll do that. Make a guess. So So I'm going to make a guess. Here's an anther. Down here, there's an anther and a fine. So maybe it's the fine that's different. Here's a bractic and a fine. Okay, so if I took the fine away, that would make sense too. But let's double check. So we have bractic, fine, and then anther. And so if we do a double cross, anther will still be wild type, and the fine will now be a wild type. So that would make sense. That would be the bractic, and the same thing is going to be opposite. So instead of a B, we get a wild type. Instead of two wild types, you get F and A, and those do match. So we know that that's the correct order. Now we want a linkage map. So we can bring these across. These two are going to be the double crosses. That's 2 and 3. And we're going to put that in both of them. So now if we do another cross where basically the A is flopped from the double cross, and it's opposite, you're going to get 20 and 26. Add them all together, you get 51. There was 1,000 progeny there. So 51 divided by 1,000 times by 100, you're going to get 5.1 or 5.1 map units away. Do the same for the other numbers. So these are the double crossovers, leave them the same. And then just swap over the B's. So you're going to get all wild type. That's going to be 98. And then all mutant, which is 87. Add them all together. You're going to be getting 190. Now 190 divided by 1,000, the total progeny, times by 100. It's going to give you 19. So 19 map units away. Determine an interference value. So divide the numbers by 100 each and then times them together. So 0.0069 and times that by total progeny, 1,000. So you get 9.69. Divide the actual, it's going to be the 2 and the 3 together. So 5 divided by this 9.69 is going to give you 0.515996. One subtracted with that, it's going to give you this, or about 48.4 interference rate. Now this last question, a little different, a lot less workout. So let's go through this. So suppose a cell has genotype A, little a, B, little b. Assume that the two A's are separate homologous chromosomes from the B and B. So what are the genotypes of the gametes produced by the cell? And what is the frequency? Well, this is just classic Mendelian genetics. So we could get either way. So we can get A with big B or little b, and then small a with big B or little b. So 4 ways, they're all going to be totally even. So capital, 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 lower, lower, capital, lower, lower, all 1 to 1 to 1 to 1. Next one, suppose a cell is the same genotype. In this case, A is linked to B, and A is linked to B. Assume that crossing over doesn't happen. So if A is connected to B, that means whenever you have a capital A, you're always going to have a capital B and lowercase a with a lowercase b. So you're going to get a 1 to 1 ratio, and it's just those two. So then supposing the same situation up here, but this time we do have crossing over, that would allow us to get these two, not heterozygous, hetero something, but where you get the capital A, little b, or lowercase a, capital B. And that's where you get those. But it's actually in question D where it says, well, what if there's 7.8 units away? So what would the percentage be? So you copy those down right here. And if you think about it, every time you have a splitting away, you're actually in 2 chromosomes. So there's actually 200% total. And if each, but there's, because half of them are just matching the other ones. So if we just focus on the first ones, we can figure out the second one. So if you ignore this section, you have AV, and then, so we're just looking at just the big A. Question is, will it be with the B or the little b? And so this is saying if there's 7.8 map units away, that 7.8% of the progeny will be little b. So you get the 92.2 and the 7.8 added together gets the 200. But if we divide everything by 2 to get to the 100%, because that's the percentage we like, you get 46.2, 3.9, 3.9, and 46.2 as all of the possible answers. And that is the end of the questions. So let me save that, and I hope you enjoyed it.